Hi, everyone, and welcome to today's lecture on protein structure. Now, a lot of this should be review for you, but it will help build us into the upcoming lectures and help you better understand some of the more complex material that's to come. So, just a reminder, proteins are macromolecules, and that's another way of saying that they're polymers made up of little monomer subunits. And for proteins, those little monomer subunits are amino acids. Now, when you have less than 40 amino acids making up a protein, we call those an oligopeptide. The spelling will be on the chalkboard notes on the next slide. And whenever you see oligo, OLIGO, in front of a word, that basically means short. So, for instance, when you work in genetics or biochemistry with nucleotides, so things like PCR with DNA, and you order oligonucleotides, those are very short pieces of DNA. And so oligopeptides are very short pieces of protein, so less than 40 amino acids. And we'll see that term oligo come up in later lectures as well for other molecules too. Now, when we talk about proteins, a big aspect of their abilities and their function is how they are folded. And ultimately, what dictates that folding is the R group of their amino acids. And so we're going to go through in the upcoming slides a reminder of what the amino acids are. But when you look at the side chains or the R groups of amino acids, that's what ends up giving proteins their different folding abilities. So, for instance, this portion here with the carboxyl side and the amine side will be the same for each amino acid and the alpha carbon and hydrogen, but then the R group varies from each amino acid. And, those interactions, whether it's hydrophobic, hydrophilic, positive charges, negative charges, all of that can affect folding. And also if they're aromatic, big and bulky. So now, as I was just mentioning, a reminder of amino acid structure. All of them will have the amino group, the NH2. They'll have the carboxyl group, which is the carbon and the two oxygens and hydrogen, and the alpha carbon, the hydrogen attached to that. But then it's that R group, that side chain that differs from each of the amino acids and really gives it the powers, the abilities, the folding interactions and all. Now, when we talk about amino acids, keep in mind there are 20 amino acids in nature. And here we have those 20. I know it's a little tough to see on this slide. I then, on the next slides, have additional pictures to help you visualize. I just wanted you to be able to see the individual letter abbreviations. So for each amino acid, you have the three-letter abbreviations, and then you have the single letter abbreviations. And for this class, I want you to make sure that you know the single letter abbreviations because that's very important. So I never make you memorize the structures of the amino acids or the chemical structures or anything, but knowing the individual letters is very important to be able to understand later topics and when you're reading different, review articles, research papers, or you're interacting in the medical field and research field. Those letters abbreviations come up. So I want you to know the letter abbreviations, and I also want you to know the categories. So the polar ones include these guys here. And so for the single letter abbreviations, you would make sure to know that polar ones are S, T, C, N, Q, and Y. And then each one here, you have the letters as well. And I'm going to write them on the chalkboard slide in the next spot. Okay, so now this is the chart that I really wanted you to see as well. So it has the same information as the previous one. It's a little clearer for you to see, not as blurry, but I like that that previous one gives you some little tricks to memorize the categories and the single letters. Now, what I want you to know on these is for each amino acid, I want you to know what category it falls into. So, know which ones are nonpolar, which ones are polar, which ones have a negative charge, and which ones have a positive charge. And again, those were also listed out on the previous slide. Also, please know the three that are aromatic and what aromatic means. So if you look at any of these, for instance, tyrosine is over here, phenylalanine is over here, and tryptophan is over here, you notice that they have the big bulky ring structures. Now, the reason why I make you memorize or know what categories amino acids are is that defines function later on. That defines how proteins will fold. So if you know that a protein, for instance, has a lot of aromatic amino acid residues, that's going to greatly affect its ability to fold or pass through certain channels or pores and whatnot, because those are big, bulky structures. If it has a lot of negative charges, so it has a lot of aspartate and glutamate residues, well, that's going to make it very attracted to any positive residues. And that's going to make it, fold in a certain way, interact in a certain way. So it's very important to know nonpolar and polar in terms of hydrophobic and hydrophilic effects. Hydrophobic nonpolar residues will want to be hidden away from water internally, whereas polar hydrophilic residues will want to be on the outside of proteins when they fold and whatnot. All of these things are important to the ultimate function and folding and just abilities of a protein. So again, please make sure to know the individual letters. So if I ask you which of the amino acids is represented by K, for instance, you should be able to tell me which amino acid that is. And so just to point it out over here, that would be lysine. And I could do that for any of them. And then also I could say which of the following are nonpolar, which of the following is polar, positively charged, negatively charged. So make sure to know those points. Now, like I mentioned a moment ago, the different categories of the amino acids affect what they can do, where they end up on proteins, where what abilities they have and whatnot. And so I want to point out over here, we're going to talk about hydrophilic amino acids and the common covalent modifications that can occur with those. So I want you to know the term kinases. Kinases will come up in many of the metabolic processes that we study in this class. So when we go over glycolysis, the citrate cycle, all of that, we're going to keep seeing kinases. What they do is they add a phosphate group to a protein. I mean, yeah, to a protein. And so you see here, kinase is working, adds a phosphate group. Kinase in this direction adds a phosphate group. And you'll notice that where does it get that phosphate group from? From ATP. So ATP is the donor that gets targeted to get that phosphate group. Again, it's usually going to occur with these two here, serine and threonine. So when you hear kinases adding a phosphate group, usually it will be to serine or threonine. Now, what you notice in these figures is kinases add that phosphate group, but in the reverse direction, what's removing it, well, the opposite of the kinase is the phosphatase. Okay, so circle star highlight those two terms. Kinase add the phosphate group, phosphatase removes it. Because in biology, you always need opposites. You don't want to have something able to be done without being able to reverse it. Okay, So now I want you to, as my famous motto says, circle, star, highlight this slide. I want you to put a bunch of stars around it and really emphasize in your notes that cysteine is a very, very important amino acid. It can do something that other amino acids cannot, and that is it can form disulfide bonds, which you see here, disulfide being two sulfur groups. And the reason it can do that is because it has these thiol groups, which is the SH that you see here. And so it can link together. Now, that's very significant that only cysteine can do that because that basically gives a stabilizing interaction in folded 3D structure of proteins. It improves rigidity and preserves stability. Okay, so whenever you see cysteine mentioned in proteins or you see disulfide bonds mentioned, please keep that in mind for stability and rigidity. Okay. And cysteines are also important because they're able to chelate metals. So that means, bind to them or attach to them, attract them, such as iron. And we're going to see that talked about in later lessons with heme. But for now, I want you to really emphasize the thiol groups being able to form disulfide bonds and that increases rigidity and stability. And we all need a little stability in our lives. So now we get to some of the not so fun stuff. Things like isoelectric point. And what the isoelectric point is basically the pH at which an amino acid is neutral. Okay, and so you see that here, net charge of 0 for an amino acid. They call that the isoelectric point, that pH that you'll find that. And you'll notice that at a pH below the isoelectric point, the charge that the amino acid will carry is a positive charge. Okay, so pK1 over here, you see that's a positive charge over here. And then you see that above above the isoelectric point, those pHs, the protein will carry a negative charge. Okay, so negative above, positive below the isoelectric point. Now, when you see terminology associated with this, the terminology is cation, zwitterion. I love that word. So cool. Makes you sound fancy and classy. Zwitterion and anion. Okay? And so what that simply means is cation. Think of a cat and how cute they are. And oh my God, I love them. So that's a positive thing. Unless you're allergic, I am so sorry. So cation simply means positive charge. Anion, you can circle and underline the N of anion to say negative charge. Okay? So when you think of that, that's cation is the positive charge that we mentioned a moment ago over here. Again, at a pH below the isoelectric point. Uh, Anion is the negative one at a pH above the isoelectric point. And at the pH of the isoelectric point, that's when you have a zwitterion, which simply means it's an electrically neutral molecule. It has a positive and a negative group on it. Okay, so zwitterion is when you have positive and a negative group. And that's found at the isoelectric point pH. Now, when we're talking about amino acids and the formation of proteins by, linking together those monomers, the amino acids to build the polymer, the protein, that tells you that you have to form peptide bonds. The process to do that is called condensation, or sometimes you hear it called dehydration synthesis. So you'll notice that you are removing an H2O group. So you're removing H2O group to link together two molecules or two amino acids. Whereas the opposite, when you are breaking things apart, breaking the bonds, that's hydrolysis. And so lysis tells you that it's breaking, like the cell breaking. So hydrolysis, you're using water, the addition of water to break apart a bond. And so you notice that water is very important in these reactions. And so just make sure to be comfortable with that terminology. Now, when we are talking about proteins and amino acids, I want to draw your attention to the genetic code. And yes, I love the mention of anything genetics. DNA is my baby. And so when we talk about the genetic code, you'll notice that it has 64 different codons. Okay, a codon being a set of three. And so you'll either see it written as the DNA codons or the RNA codons, and the RNA ultimately coding for the amino acids that you're going to see. The 64 codons have to code for 20 amino acids. So, what that tells you is that it is what we call a degenerate or redundant code, meaning that you'll notice multiple different codons can code for the same amino acid. So, over here you have two different codons coding for phenylalanine. Over here you have four different codons coding for serine and so forth. And you also have three stop codons within here too. Okay, so 64 different codons coding for 20 amino acids. This tells you it is a degenerate code and that there's multiple codons making up the same amino acids. Once you have all those amino acids put together and you are going to have your protein structure, I just want to remind you of the four levels of protein structure. You have primary, secondary, tertiary, and quaternary structures. The primary structure is simply the sequence of amino acids. Secondary is when those amino acids fold into either a single alpha helix or a single beta sheet. You then have the tertiary structure, which is where you have multiple alpha helices, multiple beta strands or beta sheets interacting with each other to form a globular protein. The very last structure is quaternary, where you have multiple of those subunits of globular proteins interacting together. So you end up with a multi subunit quaternary structure. Now, to give you a little more information on some of those levels of hierarchy, I want to draw your attention to tertiary structure examples. The first one here is that you can have a four helix bundle. And so, as the name suggests, you only have alpha helices present when you have a four helix bundle. The second one is a Greek key fold, which you see over here. These are all, all of these are representing the Greek key fold. That now will consist of four or more beta strands, and they're linked together to form a beta sheet structure. But again, this one now is only beta sheets. So the first one is only alpha helices. This one is only beta strands. Then you have the Rossmann, which you notice you're now alternating beta and alpha structures. Okay. And what you see here occurring, so you have beta, beta, and then again, beta, and then alpha, beta. That's because you have two distinct regions of alternating beta and alpha. That's different from the very last one over here, which is the Tim barrel fold, which also has beta and alpha alternating, but it's all one structure. There's not two distinct regions, okay? So you have four helix bundle, all alpha helices, the Greek key fold, which is this ABC over here, which is all only beta strands. Then you have Rossmann, which is two distinct regions made up of alternating beta and alpha. And then you have the Tim barrel fold, which does not have distinct regions. It's all one set of alternating alpha beta. Now, those were fairly simple tertiary structures. I want to show you that more complicated versions do exist. Because, like we showed before, proteins, they fold in all kinds of ways. They then end up with quaternary structure after that, and also can get complicated, kind of like life. And so, one of them that I want you to know is the firm domain, but spelled with an E instead of an I. And the firm domain you see over here, it has three distinct structural components. The first one, the F subscript A component, you notice that it's mostly beta sheets within there or beta strands within there. And you got the one little alpha. The F subscript B over here, that one instead is only alpha. And then back to FC is similar to the FA in that it's mostly beta again. Okay, now what's really important about the overall structure, which you see over here, we see disulfide bonds. And what did we say before about disulfide bonds? Whenever you think of that, think of stability, rigidity. Okay, so this tertiary structure is stabilized by disulfide bonds, and which amino acid is going to form those disulfide bonds? Well, that's cysteine. Remember cysteine. And you also, because it's cysteine, we said not only does cysteine form disulfide bonds, but it can interact with metal ions as well. So you'll see that also occurring with this kind of structure. That then brings us to quaternary structure and the terminology that you'll see with this. Now, with quaternary structure, we said that you are going to have multiple subunits put together now to make a big overall protein or polypeptide. So, some of the terminology I want you to know is having a homodimer versus a heterodimer within the quaternary structure. If you think, a lot of times people think of homosexual and heterosexual to remind themselves of same versus different. So, homodimer means that you will have identical subunits, whereas heterodimer tells you that the subunits that you have there are different subunits. And then sometimes you then beyond that see trimer, tetramer, hexamer, tri being 3, tetramer being 4, and hexamer being 6. Okay, so you have 3 subunits, you have 4, you have 6. And when you are looking at this, the way the questions might be phrased would be, for instance, what is the quaternary structure of a hexamer made of 3 different homodimers? So this first term that you see here is going to tell you how many subunits you should have in total. And so, for instance, when you look at these examples, this first one has 3 subunits, A, B, and C. This second one has 6 subunits. That's what the little subscript number is. So there's 6 subunits. They are all subunit A. Okay, 6 copies of subunit A. This one tells you you have 3 different subunits, and in total you have six different subunits in total because you have 2 plus 2 plus 2. Okay. Out of those 6 subunits, 2 are A, 2 are B, and 2 are C. The last one, 3, 3, and 3, tells you you have a total of 9 subunits. Okay, 9 subunits. And the first 3 are the same. They're all A. The second 3 are all B. The third 3 are all C. So go back to the question, and it's telling you a hexamer. So ask yourself how many total subunits I have. And then it says 3 different homodimers. Okay, So homodimers, before we said you have two identical subunits, okay? So when you see homo, that tells you same, identical, dimer tells you two. If you had homotrimers, that would tell you three identical subunits. So for instance, A subscript 3 would be a homotrimer. If you have A subscript 2, that's a homodimer because you have A and A are identical, two identical subunits. A subscript 3 is a homotrimer because you have A, all the same, three subunits that are the same. Okay, so now what I want you to do in the Remind app, take a picture of this slide, and I want you to send me the answer to this question and this one. This one's telling you a hexamer composed of two homotrimers. Okay, so the first part tells you how many total subunits. For instance, over here you have three subunits total, six total, nine total. So hexamer, for instance, would tell you six total. And then the second part tells you how they're arranged. Okay, do you have all the same? Do you have different? Okay, and so again, an example of a homotrimer would be A subscript 3. Okay, the moment that you then have a second homotrimer, then you have B subscript 3. Okay? Heterodimers would be, for instance, A, B. You have two different letters, um, two different subunits. Okay, so I want you to answer these two questions in the Remind app, giving me a picture of this slide. If you do not understand how to answer them based on what I just said, tell me that. Send me the picture and say, I don't understand this. Can you show me how to do each of these individually? Okay, because I first want to see if you're able to do them based on just the terminology. And if you can't, I will guide you through them. Please do not copy off of anyone else. Don't ask your friend for the answer because you're not getting graded on this. I do not care if you tell me I don't know how to do this. Can you show me how to do it? Okay. I just need to know one by one where each of you stands so that I can help you so that you get it correct on the exam where you actually are graded. So please help yourself out by not trying to cheat on this. This is not graded on whether you get it right or wrong. It just matters that you send it to me and we work together one on one to go through these problems. Okay, so now Now that you're comfortable in terms of the possible structures and some of the terminology that we're talking about, I want to bring your attention to what are called fibrous proteins. These are multi-subunit complexes that you have very abundant in nature. The three examples that I want you to know are keratin, silk, and collagen. Okay, nice and strong structures, very valuable structures that we're going to see in later chapters. The first one, keratin, is what you see here in this first AB figure. It's made up of fibrous coiled proteins of two alpha helices wound together. And it's linked by disulfide bonds. So you see the disulfide bonds here linking together the coils, which tells you that keratin is nice and rigid and strong. You then have the silk, which is this A, B, and C here. That's also a fibrous protein, but now this one is consisting of almost all beta sheets, which collectively ends up giving silk its strength. And then the last one of the examples is collagen over here, which is made up of three intertwined left-handed helices up here. And again, these are structures that will come back in later lectures. Now, since I am in the biology field, I like to kind of bring up medical connections within these lectures. This one is a pretty cool historical medical connection between what we've just been talking about and the medical field. So, if you've ever heard of scurvy, that is basically a disease where you end up having connective tissues in the skin and blood vessels that break down. And this is because of structural defects in collagen that we just saw on the previous slides. Okay, now I then asked, why do diets deficient in vitamin C lead to scurvy? So if you ever heard, Lyme being able to protect people from scurvy, that's because vitamin C is a very important coenzyme. So coenzymes are needed to activate enzymes and help reactions occur. So vitamin C is a coenzyme for the hydroxylation of proline to what we call 4-HYP, so hydroxyproline. So you need vitamin C for this reaction to occur, and this 4-HYP is required for the strong non-covalent interactions that make up the collagen helix. So vitamin C is the coenzyme for the reaction that helps to put together the collagen helix, okay? And so if you do not have enough vitamin C, you do not have enough of the collagen structure, you end up having issues with connective tissues and in skin cells and in blood vessels that break down, and you end up seeing things like this, which is just nasty. So please make sure to have your citrus and vitamin C. I forgot to mention on the previous slide, you don't need to write it down on your notes, but just in case you're curious of some of the symptoms that you see with scurvy. I know I showed the picture with the teeth coming out and nasty gum effects, but ultimately symptoms can also include at first just feeling weakness and tired and sore arms and legs, kind of like you went to the gym a little bit too much, which I know there's no such thing, but yeah. Uh, and then it progressively gets worse. So you start seeing decreased red blood cells. You have the gum disease that I showed in the picture, changes to the hair, bleeding skin, and it can even get so bad that you end up with poor wound healing, even have personality changes, and finally you could get death from the infection or bleeding. So since I know a lot of you want to go into the medical field, even though you don't think that you see scurvy as much nowadays, in some of the third world places, you actually do see it happen. And, with people always going on fancy, social media craze, diets, and all, it's valuable for you to know these kind of things to look out for, even though we may think, oh, we got rid of that kind of disease. And we see that now with, also people spreading anti-vax stuff on social media, and now suddenly diseases that we thought we got rid of because of vaccinations, they're popping back up too. So always important to know these things if you're going into the medical field. Then I want to point out examples of globular proteins that are very, very important to us. These are immunoglobulins. And if you've ever heard IG or immunoglobulins, that's a fancy term for antibodies. And so when I say what are these, they are antibodies. The folding is an all beta structure that includes the Greek fold. So you're seeing all beta structures here. And what helps maintain their stability? Well, what did we keep saying? I want you to think of when you hear stability and rigidity, think of disulfide bonds, okay? So they have, over here, you can see disulfide bonds helping to keep those subunits together, okay? So they are linked by disulfide bonds and the making up the heavy and light chains, keeping them together. And because of those bonds, when you mildly treat these Y-shaped antibodies with a protease, which would, break apart proteins, these guys will still maintain structure and function within the individual subunits because they're held together by those strong stabilizing disulfide bonds. And so you can end up with what we call Fab and FC fragments. So you see here, cleave with a protease, you still have the Fab and you have the FC fragments. Now the Fab, you'll notice it's this upper portion, the light chain here, which has the antigen binding sites. That's very valuable to us in the medical field because now you have mini versions of antigen binding antibodies. Okay? So they can still function to bind target antigens, but now they're much smaller, so they can easily penetrate tissues. Okay, so they're very, very valuable to us in the medical field. And that's all thanks to disulfide bonds. Okay, so now we get into some of the scarier, more complex looking stuff. This is just a little more detail on protein folding. In future lectures, we'll go through exact folding as well. But this is kind of connecting some of the other stuff that we talked about to today's lesson. So the first question asks, how does thermodynamics play a role? Now, previously we mentioned that in thermodynamics, if you want something to happen, you want the reaction to be favorable, right? And favorable would be a delta G that's negative, that's less than 1, right? And so that would be, the free energy difference between the native or folded protein versus the denatured or unfolded protein. Okay, so you want the delta G to be negative. Now, when we talk about the folded and unfolded protein, when you look at this, there are a lot of different things that can basically help provide, a favorable enthalpy and a favorable kind of reaction of folding and all. And some of these include hydrogen bonds, the hydrophobic interactions, ionic interactions, or even van der Waals that we talked about. Okay, so you can have hydrogen bonds, ionic interactions, van der Waals. We also see here, hydrophobic groups mentioned. And ultimately, when you're forming bonds, that's going to be exergonic or exothermic. And so you're going to end up with a negative enthalpy that's favorable, okay? So things like hydrogen bonds, ionic interactions, and van der Waals help to provide favorable situation. Now, you may ask yourself, well, when we talked about thermodynamics and bioenergetics, we said things want to go toward greater entropy, which is greater disorganization, less order. So if a protein's folding and becoming more ordered, how is that going to be encouraged if we're not going towards greater disorder, we're going toward greater order? The answer to that is that the folding of that protein is actually a coupled system. So it's coupled with the water molecules that are around that protein. So over here, you see the water molecules and then the water molecules over here. And basically, what happens is as you fold a protein, okay, water allows for hydrophobic interactions with the amino acids and with the parts of that protein. And so as the protein is becoming more ordered and folding, what do you notice happens to the water molecules? They end up more disorganized, less order as they're kind of pushed around away from certain parts of that protein, the way that it's folded. So they start off very orderly around and organized around the hydrophobic groups. And then once the protein's folded, the water is now scattered about. So even though the protein is becoming more ordered, which is less entropy, the surrounding water is losing order, which is greater entropy. And so this overall process of protein folding now becomes favorable. Now, when we talk about that folding of proteins, we have been able to study that both in vitro and in vivo. In vitro, we use one of the things called circular dichroism, or CD for short, and that's basically a form of spectroscopy that measures absorption differences between the left and right-handed polarized light to look at, secondary structures of proteins, which is all a fancy way of saying that CD is a form of spectroscopy that shows the amount of alpha helices, beta sheets, and unstructured regions of a folded protein. Then we have what's called a transition curve and a transition curve midpoint. So that's what we show over here. And basically, this is a curve that helps demonstrate the amount of unfolded versus folded protein. And so what you notice is the transition curve midpoint, or T subscript M, which is over here, is going to be the temperature at which half of the, or 50% of the proteins in a protein sample are fully folded and 50% are fully unfolded. Okay, so half of the molecules are unfolded, half are folded. And what you notice in this curve is that the amount of unfolded protein increases as a function of increasing temperature. Now, why is that? What happens to proteins at high temperatures when they're hot? They denature, they unfold. So that's why you see that kind of a trend in the curve. We are also able to take a look at protein folding in vivo within the cells. And what we study there are chaperone proteins. So, chaperone proteins help proteins fold. There are two main types. There are the clamp type, which are the heat shock proteins. And then there are the chamber types, which is like this structure here, GROL and GROS. Those are the chamber types. And the difference between these two types of chaperones is the clamp chaperone proteins, like the heat shock proteins. These prevent premature folding. Okay, so you notice, as for instance, as a protein is being made, the heat shock proteins will start binding to it to prevent it from starting to fold. The chamber type, like the GROL and GROS protein complex, what that's for is once you have, let's say, a protein that became denatured, unfolded within the cell, if it goes into one of these chambers, it gives it a place to refold properly without having all the other stuff in that cell trying to interact with it, trying to bind to it, trying to force it to fold a certain way. So, the first one, the clamp, is to prevent premature folding. The chamber is to help basically give misfolded ones a place to refold. And they end up using ATP hydrolysis to help facilitate the correct folding, which you see in this structure here. Now, when we talk about protein misfolding, like you heard me mention in the previous slide, that's the last thing I want to talk about in this lecture. Up to 30% of proteins synthesized need to be refolded or degraded. And the reason why that's so important is if these misfolded proteins are not refolded or removed, they'll start messing around with the function of normal protein within that cell. Okay, they can do things like form aggregates and prevent proper function. So that then brings me to the question, what's the difference between if a misfolded protein is degraded or if it's aggregated, which are the two main options for that protein? If it's degraded, we call that a loss of function mutation. Okay, because if you degraded the misfolded protein, well, now you've lost that protein, the activity of that protein is now missing or less concentrated, and you can end up with diseases from that, like cystic fibrosis, which basically causes severe lung and digestive damage and is mainly related to problems with mucus. Aggregates, on the other hand, if a misfolded protein is forming aggregates, that's called a gain of function mutation. So now degraded got rid of the protein because it was misfolded and you lost that protein's function. Now you're getting aggregates, you're getting clumps and collection of this protein. That's going to be an accumulation, a gain of function. And you see that in things like CJ disease and Huntington's, where you have both of these are neurological and degenerative. You see dementia and death even and complications with brain function. Okay, so either way, protein misfolding is a really bad thing. Even though the cell tries to correct for it and can degrade that protein, it's still a problem because now you're missing that protein. If it forms aggregates, that's a problem. And this figure here shows you what I was talking about in terms of the examples of some of the diseases. This one here is CJ disease that I mentioned. And you can see degradation is loss of function, and this is aggregates, which are gain of functions, and both can be a problem. That then brings us to the two review questions here. Please take a picture of this slide, send me the answer in the Remind app, or if you're not in my class, you can send me an email or leave it in the comment section. The first question was, what is a transition curve used to demonstrate? The second question is asking about which amino acid has an R group that forms covalent disulfide bonds. Okay, so please make sure to send this to me in the Remind app anytime, day or night. That is it for today, even though I'm sure all you heard was blah, 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 which is just like here. Uh, and as always, if you have any questions, any concerns, just send me a picture of the slide and ask away. Thank you and have a great day. Look at that, I rhymed. All the cool people rhyme, just like Sammy and Omar on the Breaker Bros. So check them out on YouTube.